Welcome friends, myself Shoptaporni from BioLive. Hope all of you are doing very well. Today I am going to teach you about a very important topic of biotechnology, the pyrosequencing. So before beginning, I would like to request you to kindly subscribe to my channel by pressing the bell button given below. And if you like this video, don't forget to like it and share it with your friends. And if you have any doubts regarding this video, you may leave a comment below in the comment section. You may also follow me in my Facebook page, Facebook group or in my Instagram profile. The links are given in the description box below. So okay, without further delay, let's begin. So what is pyrosequencing? From the name, it is evident that this kind of sequencing depends on the release or liberation of pyrophosphate. Okay, and how does this occur? We will see that in a little bit. Okay, say this is your target DNA or the template DNA whose sequence you don't know. Okay, you have to find out the order of DNTPs here. Okay, so how will you find out that? For that, first of all, you need to amplify it through PCR. Okay, and after amplification, you can do the pyrosequencing process by either solid phase reaction or by the liquid phase reaction. For solid phase reaction, you have to attach the template DNA or the target DNA to a solid base. Okay, and what is that base? That base is usually the streptavidin coated magnetic beads. Okay, so for that we actually biotinylate our PCR primer. Okay, we add a biotin molecule to the PCR primer so that whenever the DNA gets amplified after the PCR, it can be attached to the streptavidin molecule present on the magnetic bead, and the DNA template can be bound to a solid surface. Okay, and then you can do the further processes of the pyrosequencing. And for liquid phase reaction, the DNA should be present in the solution. Okay, so after you have bound the DNA in a solid surface or the DNA template is in the solution, you have to denature it. You can denature it by adding the alkali solution, or you can also denature it by heating. After denaturing, you have to add a sequencing primer to the single-stranded DNA template. Okay, so that the DNTPs can be added to the 3' OH end of the primer. So this green one is our primer, and this is the 3' OH end of the primer where the subsequent complementary DNTPs are to be added. Okay, so we know that with this 3' OH group, the next DNTP will form a phosphodiester bond with its 5' phosphate groups. There are 3 phosphate groups. This is alpha, this is beta, this is gamma. And this 3' OH group will attack this bond. Okay, and it will form a phosphodiester bond with the next DNTP. And the next DNTP will be attached here. And these beta and gamma phosphate groups will be released as pyrophosphate. This one is our pyrophosphate, PPI. Okay, and in the pyrosequencing process, this PPI will get converted into ATP by reacting with APS. APS is the adenosine 5' phosphosulfate in the presence of ATP sulfurylase enzyme. Okay, now how can you detect this PPI? Okay, this ATP will get converted to light in the presence of luciferase enzyme, which reacts with luciferin and converts it into oxyluciferin. So this light can be detected by a machine known as CCD. CCD is the charge coupled device. Okay, and this charge coupled device will show the light, okay, in the form of a peak in the pyrogram. Okay, if the right kind of complementary base is added here, it will show a peak like this. Okay, and the height of the peak is directly proportional to the number of DNTPs added here. Okay, so we will see it in a little bit. Okay, after that we have to remember the main funda of the pyrosequencing. Here, DNTPs are only one single type of DNTP is added at a time. Okay, what does that mean? That means first, let's see, this is the order of DNTPs to be added. First DATP will be added, then DTTP, then DGTP, then DCTP. Okay, that means first we will only add DATP. Okay, after adding the primer sequence and all other enzymes like ATP sulfurylase, luciferase, and other substrates like APase, luciferin, we will add only one single type of DNTP. It can be either DATP or DTTP or DCTP or DGTP. Let's say first of all we will add DATP. And if there is the complementary base of DATP, that means there is the complementary base of adenosine there, that means there if there is T, then only the adenine will get attached here. And if there is no T, then ATP will not get attached here. So as here the T is present, DATP will get attached here and will form the complementary base pairing, and as a result the light will be formed, and this light can be seen in the pyrogram as a peak, okay, as a peak of A, okay. Next we will add DTTP. So DTTP will complementary base pair with A, okay. But here see the next base is not A, next base is G. So the DTTP will not get attached here and no light will be formed, okay. So to remove the DTTP from the solution, we need another enzyme known as apyrase, okay. Apyrase will remove all the wrong DNTPs, and it will also remove extra DNTPs from the solution, okay, so that there is no overlapping of DNTPs in the next step, okay. So DTTP cannot bind with G. Next we will add DGTP. Guanine also cannot bind with G, so there will be no peak here in T or G, okay. Next we will add DCTP. Cytosine will bind with G, so we can find a peak here in the C region, okay. Next again we will add the DATP, okay. See here next base is A. There are two A's, okay. So obviously adenosine will not be attached here. Next we will add DTTP. Two DTTPs will be attached here, and you can see here is a peak at the T. There is no peak at A. There is peak at T, and this peak is longer in height than these A or C. Okay, that means here we can prove that the height of the peak is proportional to the number of DNTPs to be added. As here two T's are added, the height of the peak is more than these peaks. Okay. Then we will add DGTP. See here C is present, so obviously G will attach here and will give a peak. Okay. Then we will add DCTP, but the next bases are T. Okay, so obviously C will not attach there, so there is no peak at C, but there are peaks at when DATPs will be added. See here DATPs are added and 3 adenosines will be added here, and the peak is longer in the length, okay, height, okay. So as 3 A's are added here, okay. In this way, the process will run, and you can, by examining the peaks, you can assess the sequence of or order of DNTPs present in your template DNA or target DNA, and that is how pyrosequencing works. Okay. Now here, another thing is that you cannot add simple DATPs here, okay, in this reaction. Why? Because if you add DATP, it can give the negative result or false positive result. Actually, it can give false positive result. Say there is no complementary base here of adenosine. Okay, say there is G or C like this. Okay, but in that case also DATP will react with luciferase and will give the light. So that kind of peaks are for negative peaks, okay, or false positive peaks actually. So to avoid this kind of situation, to avoid the reaction of DATPs with luciferase, we actually use DATP alpha A's. That means D-adenosine alpha thiosulfate, okay, instead of normal DATPs, okay. So this is the everything about pyrosequencing, the basic of pyrosequencing. I hope this is now clear to you. Okay, so you have to simply remember that only one type of DNTPs can be added at a time in case of pyrosequencing, and this is the main funda of pyrosequencing. Okay, I hope you have got it crystal clear now. Okay, So if you find this video useful, don't forget to like it and share it with your friends. I will come up again with another video of sequencing in my next video. Okay, so for the time being, thanks for watching and happy learning.